thank you. I want to thank the organizing committee as well for inviting me to come and speak on the panel. So thank you very much. My name is Laura Peak, and I am a principal scientist and assistant lab director for Oncimmune. And as long as I stick to my slides, I can do this in 10 minutes. So if I start to ad-lib too much, I'm probably going to go over. So Angela provided a really great background to clinical laboratory medicine, and I think you'll see in my talk the direct application of clinical laboratory medicine in light of a particular test and how it fits into a lab world. So Oncimmune is a clinical laboratory. Unlike large reference laboratories, we just perform a single proprietary blood test that we developed. So that's what I'm going to talk you through. So in my role, I oversee all the laboratory operations, so both high complexity clinical testing as well as all the R&D that we continue to do. I was responsible for developing our quality system. So you'll see I was the first employee hired, so I have done a lot of things. I've also been involved in all the method development, particularly with new batches coming in, how we qualify those to make sure that they meet our quality criteria. On a daily basis, I perform complex data analysis, and I also managed an audit of clinical outcomes for 2,000 patients. So these were patients who were tested by our test, and we sent out questionnaires to physicians to find out whether the test was really working in clinical practice as we intended for it to be used. So we asked the question of whether or not the patient who had the early CDT lung blood test, were they later diagnosed with a lung cancer? And I'm starting to ad-lib too much, but I'm pretty passionate about some of this stuff, so forgive me. I'm also responsible for the regulatory compliance for the laboratory, and I'm involved heavily in strategic planning for the company. And then most recently, I've actually been doing a lot of business development, talking to physicians, talking to lab directors of clinical reference labs, trying to create a relationship whereby they might market our test to allow another sales outlet for the test. So my journey really started my junior year of high school when I took chemistry and really became interested in chemistry. It was challenging. I became really passionate about the subject, and I thought, okay, how can I use this and make myself happy? So I definitely knew all along, since I was little, that I wanted to do something to help people. And so how can I help people while utilizing chemistry? And I thought, well, I could develop drugs to cure disease. That'll be a good way to do it. So when I graduated high school in '98, I went on to Baker University and pursued a bachelor's degree in chemistry and got a minor in math along the way. While I was there, I had the opportunity to do some internships. So I interned at Aventis Pharmaceuticals in the summer of 2000, and then during the January term for 3 weeks in January between Christmas and the spring semester. And at that time, I was in the quality control raw materials lab. I quickly became very proficient in the tests that I was performing there and really didn't feel challenged. So I thought, well, grad school might be in my future. So the next summer, in summer 2001, I did an undergraduate research opportunity at KU in the pharmaceutical chemistry department. And then in the summer of 2002, I went back to Aventis and did an internship in the dissolution lab, again, quality control lab. So again, I became very proficient, didn't feel challenged, and I thought, okay, grad school is my next step. So in 2002, I joined the pharmaceutical chemistry department to pursue a Ph.D. here at KU. My advisor was Russ Midoff, and I was fortunate enough during the 4 years of my graduate school to have a Madison and Lila Self graduate fellowship. So they supported me for the 4 years, and the development program that goes along that was extremely beneficial to my career path. So my doctoral research specifically focused on developing a high throughput approach to pre-formulation and formulation development of recombinant protein therapeutics. And in developing that high throughput method, I had the opportunity to work on vaccine candidates for anthrax, malaria, and ricin toxicity. So during this process, we were adsorbing these proteins to the surface of aluminum salt adjuvants, and I had read a lot in the literature about protein surface interactions causing destabilization of the protein. So I thought, well, here we are creating a drug formulation where we're adsorbing a protein to a surface. What's happening to these proteins? Are they destabilized? We're trying to stabilize them in solution, but then we're sticking them on a surface. So what's happening? So I developed techniques to investigate that surface interaction, and in fact found that they are destabilized on the surface of aluminum salt adjuvants. So then I got to thinking, well, we really don't know how protein vaccines work in terms of eliciting an immune response. There are numerous mechanisms proposed in the literature, and I proposed another mechanism, and that is that the unfolded structure of the protein may actually make the protein more susceptible to proteolytic degradation in the antigen presenting cells. and may in fact help facilitate that antigen presentation. So after grad school, I went on to do a postdoc in Corey Berkland's lab at KU. He has a joint appointment in pharmaceutical chemistry and chemical engineering. And so I had just, in my doctoral work, done a lot of work in formulation, pre-formulation. And so the natural next step was to look at drug delivery problems. So Corey's work was in the area of drug delivery, and specifically I worked on an inhaled drug delivery system to the deep lung, which consisted of self-assembled flocculated nanoparticles. And then I also worked on a tissue engineering project looking at encapsulation of growth factors in PLGA microparticles. So then, what was I going to do? So the job search continued, and really Corey was extremely supportive in terms of encouraging me to continue my job search because he knew I wanted to stay in the Kansas City area. My husband had a great job downtown, my family's all nearby, and all of the pharmaceutical companies are on the coast. So left me with a real dilemma. And one day I was walking in the multidisciplinary research building on Lawrence campus in the hallway, and I ran into my doctoral advisor, and he said, "Hey, Laura," he said, "the CEO of Oncimmune is going to be here later today. Would you like to meet him?" And I said, "Well, yeah, sure, I'd love to meet him." So I met him, but the meeting him turned into a 2-hour interview, and they were looking for a scientist to help start up the lab. And he called me back the next day, offered me the job, and again, networking is key. It's all about who you know. So in April of 2007, I joined Oncimmune. I was the first employee hired for this new startup company. So my first day on the job, let me just say, at that time we were subleasing a corner of lab space from an existing laboratory in Lenexa. So my first day on the job, the CEO, who's based in Atlanta, isn't there. So I think, okay, well, we're going to tackle this. So I go and I ask the lab manager, okay, can you show me our space, first of all, because I didn't even know where we were. And this is essentially, this isn't the exact space, but this is essentially what she took me to. And I'm like, okay, yeah, I never have taken a class in setting up a laboratory. Have any of you? Have you? That's impressive. So I tackled it, and somehow or another, I set up a laboratory. And this is, so we subleased the corner lab space for about a year. And in the spring of 2008, we moved into our own laboratory that we built out to our specifications in DeSoto. And these are just some pictures of our lab now. So as I reflected back and thought about, okay, what did I learn in grad school that helped me to be successful? Because clearly, somehow I did it. So I started thinking about some of the skills that I learned in grad school. And I came up with, to utilize my resources. Utilize every single resource around you at all times. Ask lots of questions. Certainly resourcefulness. If you don't necessarily have the tool you need, you figure out some other way to do it. Creativity. Lots of problem-solving skills. Perseverance certainly plays a role. Not to overthink things, but instead just do it. And negotiation skills. So who is Oncimmune? Just briefly, I want to talk to you about what we are all about. So we are a technology company, first and foremost. So we have developed an ELISA-based platform technology that is capable of detecting all types of tumor-forming cancers. So the difference between the different types would just be the panel of antigens that we coat on the plates. So the scientific rationale behind the test is really quite simple. So as a normal cell undergoes tumorigenesis, it expresses on the surface these abnormal tumor-associated antigens. And these tumor-associated antigens are viewed by the immune system as foreign. So the immune system evokes an immune response and the production of autoantibodies. And it's these autoantibodies that we measure in our technology. So when we were thinking about, okay, what cancer do we want to tackle first in terms of developing a blood test to aid in early detection, we looked at where there was the greatest need. So lung cancer certainly is, there's a huge need for early detection of lung cancer. It is the #1 cancer killer in the US, and that's because mostly 80% of lung cancer is diagnosed once it's metastasized. The 5-year survival rate's only 17%, and 56% of the patients diagnosed typically die within a year of diagnosis. Certainly there are a lot of lung cancer cases diagnosed annually, over 220,000, and smoking is definitely one of the main factors contributing to lung cancer, and there are about 42 million smokers in the US. So the same number of people die each year of lung cancer as die of kidney, liver, prostate, breast, and colorectal cancers combined. So we developed early CDT lung to address that problem. So did I take the typical road to get to where I am? Probably not. But I followed my heart. It made me happy. It's a very personally rewarding career to know that I'm helping to save lives through detection of early stage lung cancer. Joining a startup, I gained a ton of experience that I definitely would not have gained otherwise if I would have joined a large company. I've had the opportunity to wear many hats. I'm involved in HR. I mean, some of the hats I like better than others, but, and then some of the things that are necessary when you do join a startup, just to make you all aware if any of you are thinking about a startup, is that you must be a multitasker, you must be able to work under pressure, and most importantly, you must be a doer. You can't just sit back and ride the wave. They're expecting you to do that, so you have to figure out a way to do it. The cons are probably my degree specificity. So I have a Ph.D. in pharmaceutical chemistry, and now I'm working in a clinical laboratory setting. So it doesn't really apply, but certainly the protein characterization techniques that I learned in grad school were what were most attractive to Oncimmune when they hired me. They did definitely take a leap of faith with me, and I am grateful every day for that. And then also the second con I've listed is that I do lack the breadth of clinical lab experience. So I have tried and failed at getting board certification to become a clinical lab director like Angela spoke about. And what it came down to is I just don't have the hands-on experience that I need to pass that board exam. So again, getting as much experience, knowing up front what field you want to go into and knowing what qualifications it's going to take to get you to the top, pursue that path. That's probably the better path than the path I've taken. So with that, I'd like to thank you.